Well, hello and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled Innovation in QT Strategies, Leveraging Early Stage Safety Methodologies and AI to Maximize Your Opportunity for a TQT Waiver. My name is Corey Stanton and I'll be your X-Talks host for this event. Today's presentation runs for approximately 60 minutes and the webinar includes an interactive Q&A session at the end. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation, and we'll try to attend to as many as possible. If you're joining us via GoToWebinar, you can use the questions chat box, which is located in the control panel on the right-hand side of your screen. And if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. And if you're viewing this presentation from our LinkedIn Live event, you can submit your questions using the comments tab on LinkedIn. Please note that all audience members are in listen-only mode and that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions that generate high-quality clinical evidence for their pharmaceutical, biotech, and medical device partners. They offer comprehensive evidence generation solutions that combine eCOA, cardiac solutions, medical imaging, precision motion, and respiratory endpoints. Since their founding more than 50 years ago, Clario has delivered deep scientific expertise and broad endpoint technologies to help transform lives around the world. Clario's endpoint data solutions have supported clinical trials over 26,000 times in more than 100 countries. Their global team of science, technology, and operational experts have supported over 60% of all FDA drug approvals since 2012. For more information, you can of course visit clario.com. Now, before we get started, let's take a moment to meet our panelists for today's webinar. Our moderator today is Dr. Todd Rudo. Dr. Rudo is board certified in cardiology, cardiac electrophysiology, nuclear cardiology, adult echocardiography, and internal medicine. He leads scientific and regulatory strategy to ensure delivery of high-quality clinical trial endpoint data. Dr. Rudo focuses on improving clinical trial rigor through innovative technologies, prioritizing patient and site experience. And also joining us today is Dr. Vik Patel. Dr. Patel is a board-certified cardiologist with arrhythmia research expertise and focuses on drug development and clinical safety across all phases. Dr. Patel oversees cardiology science and research, consulting, and ECG or Holter core lab services, and also leads the biostatistics and medical writing groups at Clario. And rounding out the panel today is Jean-Philippe Couder. Jean-Philippe has experience in the development of groundbreaking technologies in the field of quantitative electrocardiography and cardiac safety. He founded iCardiac Technologies and led the development of the telemetric and Holter ECG warehouse. And now, without any further ado, I'd like to hand the mic over to our moderator, Dr. Todd Rudo, to kick off the presentation. Dr. Rudo, the floor is yours.
Okay, great. Well, thanks for the very kind introduction, Corey. Good morning and afternoon, everyone. Thank you for joining us. As Corey said, my name is Todd Rudo. Very glad to have the opportunity to host this interesting and quite packed session today, covering innovative strategies for assessing QT risks in early development. Over the next 45 minutes, we're going to speak about how implementing the right strategy and technologies in early development can generate sufficient data to satisfy the requirements for a TQT waiver much later in development. On the screen here is our agenda. I'll start us off with a brief overview of cardiac safety and drug development with a commentary on early phase study designs as well. Dr. Patel will then take us through the history of the E14 guidance revisions, which defines the requirement for submission of QT safety data. He'll also review an ECG analysis methodology we developed called Early Precision QT, and that's an integral part of the early phase safety strategy. And then after this, we'll hear from Dr. Couder about how this EPQT methodology was initially developed. He'll also speak to how more recently we've been applying AI to assess quality of ECG data in early phase studies and to ensure that the data collected will be suitable to support an analysis per these guidelines. We do intend to leave some time for Q&A at the end, so please use the chat function as Corey mentioned, and we'll address them when we're completed. Before we do this, you can go to the next slide. I wanted to review the results of a survey that we ran in the weeks leading up to this webinar. Thanks to those of you who took the time to respond. We asked, how familiar are you with intensive QT analyses and their role in early phase studies? We had 4 answers we provided, ranging from very familiar and having done this before, all the way down to no exposure whatsoever. And as you can see here on the screen, we have a range of experience joining us today, which is perfect. The highest percentage of you have been a part of an early phase intensive assessment at some point. and then fairly evenly spread, ranging from light familiarity to no experience at all. So hopefully everyone will find this session today informative, and of course we'd welcome any follow-up afterwards. If you have further questions, our general inbox, science@clario.com is probably the best way to reach us. So why don't you go forward to the next slide. You can actually go one more. And we'll start with cardiac safety. So just very briefly to level set everyone before we get specifically into the QT world, it's an important reminder here, there is a much broader set of potential cardiovascular risks to keep in mind. For the most part, we're talking about cardiovascular risks that are associated with non-cardiovascular drugs. As you think about it, with any drug that is systemically absorbed, whether it's a pill or an IV medicine or a skin cream or an eye drop, once it gets into the bloodstream, it's going to circulate through the heart. So any drug of any type could have cardiac side effects. And those side effects can manifest in many different ways, from increased risk for heart attack, risk for damage to the heart valves, risk for what's called proarrhythmia, meaning the drug can make a patient more likely to develop a significantly abnormal heart rhythm, risk for affecting blood pressure, weakening the pump function of the heart, which leads to congestive heart failure, and last but not least, the risk for affecting the ECG in any of several different ways. So we have different tools to monitor for these different cardiac side effects. You don't necessarily deploy these all in development programs. It does depend on what preclinical risks have been identified or are already attributed with that drug class, for example. So today our focus is on the last bullet in that list there, the risk for a drug to cause QT prolongation. And why? Because we know that when a drug causes QT prolongation, the risk for that patient having a lethal arrhythmia that can lead to sudden death goes up. You can go to the next slide. So first in human studies. First in human studies are critical decision points for drug development. Once a drug candidate has been selected and the appropriate bench testing and animal testing has been completed, it's time to run a first in human study. Most often these will include a single dose study where a single dose of the drug is given to a healthy volunteer, and we measure how the drug is absorbed and how it's metabolized, and we monitor for any side effects as well. And although only a single dose is given, monitoring can occur for a week or more afterwards. And once safety is confirmed, another higher single dose is given to another group of volunteers. And then based on what we've learned from that single dose escalation study, a second study or a second part to that same study will usually commence, and that's with a multi-dose regimen. And that could be daily for a week, it could be twice daily. It really just depends on the kinetics of the drug, which were understood from that first portion, the single dose portion. You can go to the next slide. So as mentioned, here's a schematic of how this all maps out. A relatively small number of subjects are treated for this, usually 6 or 8 in a cohort on active drug and another 2 on placebo. In some cases, that multi-dose portion is sequential to the SAD. It can also be done in parallel, and that's true if the multi-dose exposures will be lower than the single dose exposures that have already been tested. In most therapy areas, the first in human is performed in healthy volunteers that are confined to an inpatient research unit. In some cases, classically oncology, for example, the first in human study may occur in patients with the disease. And although the kinetics of a drug aren't understood yet, and therefore going directly into patients does have some risk, the risk to healthy volunteers based on the safety profile of a drug may be even higher. And so the only ethical option is to go straight to patients. Next slide, please. So as I mentioned, the purpose of the first study is to understand how a drug is absorbed, how it is metabolized, and also how well tolerated it is. And we look at a range of doses and therefore a wide range of different exposure levels in the bloodstream throughout those studies. One of the important pieces of information we can also learn through a first in human study, though, is how the drug is interacting with the body. And this is called pharmacodynamics as opposed to pharmacokinetics, which is what I was speaking about before, absorption and metabolism. So an example of a pharmacodynamic effect that we can measure is how a drug is affecting the ECG. The PK information, so what we've learned, in a first in human study about absorption and metabolism, that plus some information about pharmacodynamics will often directly determine a go, no-go decision for a development program. The largest percentage of molecules that don't make it to the market will be terminated right here after the first phase of development. And the attrition is quite high for good reason. Later phase development programs with larger and longer studies are much more expensive and will be exposing many more patients to risks. So if a drug doesn't have the right profile, it's not a sound decision to keep progressing it. But, and here's kind of the key message, when such an impactful decision is being made about the program, the quality and the accuracy of the data that goes into that decision is paramount. And that's what we're going to be focusing on today. How can we ensure the most accurate and high quality data is collected from a first-in-human study to enable sound decision making? What can be done to trust the ECG data, to trust the indicators of any drug-related changes in that ECG data, and therefore ensure we understand the cardiac risks of a drug before we advance to the next phase. You can go to the next slide. So before I hand over to Dr. Patel, just a quick recap. As I said, the first-in-human, it's an initial look at how a drug is absorbed and metabolized, which is the kinetics piece of it, and also a look at safety and tolerability. We can look at some types of measurable pharmacodynamics also, so how a drug is affecting the body, or the ECG interval in this case today. Most often a first-in-human study includes a very large range of doses and exposures, more so than any future protocol will. And it's in this range of exposures really that we see the most value for engaging these early phase ECG strategies, which we'll be talking about. But without precise and accurate data, it may not be possible to detect an important signal through the noise, and that's what we're going to focus on. And so let me turn over here to Dr. Patel, who's going to speak to how the E14's guidance on the assessment of QT safety has evolved over time, and also review some of the technological advancements that have enabled a higher level of precision when analyzing this type of data. So Dr. Patel.
Well, thank you very much, Dr. Rudo. And we can go to the next slide, Corey. So the first thing I want to just go over is a historical perspective of how the E14 guidance was developed. Now, it's been known for quite some time, since the 1960s, that QT prolongation is a surrogate marker of sudden arrhythmic death, specifically torsade de points. But it wasn't until the introduction of Seldane on the market that it was seen a small but significant increase in sudden death in these patients who were being treated for allergic rhinitis, that it was found that QT prolongation could actually happen with a drug. This led to the development of the E14 guidance and release in 2005, which now mandated that rigorous QT prolongation assessments had to be done for all drugs before they'd be approved for going on the market. After the E14 guidance was released, QTc prolongation became the main reason why drugs were withdrawn from the market or stopped in development for cardiac safety purposes. And then in 2015, the guidance was further revised to include concentration QT modeling based on the results of the IQ-CRS study, because now you could assess QTc prolongation in small cohorts in early development in the single ascending, multi-ascending design. Let's go to the next slide. This slide shows a timeline of development of the E14 guidance. As I said, in 2005, the first revision was the guidance was released. In 2008, the first Q&A came out, which provided some guidance on ECG methodology and QTc assessment in gender, male and female differences, as well as the need for a positive control. In 2012, the second Q&A was released, which gave further guidance on ECG methodology and QTc assessment by gender. But it also now introduced the concept of late phase assessments of QCG, not just early phase QTc assessments. In 2014, the third Q&A was released, and this was an important update, which provided guidance that you no longer had to do a third QT study for a large biologic molecule, which really couldn't get inside the cell or really had targeting effects on the cardiac ion channels. And importantly, it also provided guidance on special populations, specifically oncologic populations where it was not safe to give a positive control or ethical to give a placebo control, just having to maintain most of the features that you could of a positive QT study. In 2015, revision 3 of the Q&A4 came out, and this introduced concentration QT modeling as another method for assessing QT risk in addition to by time point analysis. And then in 2022, the last revision came out, which provided a combined EB7 and E14 guidance, where you can now have an integrated approach to assessing QT prolongation. This meant that you could use either a double negative approach for the in vitro hERG assay and the in vivo QT assessment in the dog and monkey, and then help you make a decision to actually go to lower concentrations in the clinical study. And let's go to the next slide, Corey. So I want to go over the iQCRS study at this point. And this was released in 2015 in December. The objective of this study was to really determine if data from early phase studies with small cohorts could either replace the traditional TQT study or be used to complement it. It was designed in combination with the FDA, the IQ Consortium, and the Cardiac Safety Research Consortium. And the study enrolled 20 healthy volunteer male and female participants who were dosed with 6 different drugs and placebo. There was 5 QT prolonging drugs. These were drugs that were known to have a QT prolonging effect, and 1 drug that was known to not have a QT prolonging effect, as well as placebo arm. There was 2 dosing days in the study. On the first day, the dose was given to try to increase the QT interval by 10 to 12 milliseconds. On the second day, the drug was increased to give a prolongation of 15 to 20 milliseconds. And the ECG analysis was collected by the early precision QT methodology, which was developed by iCardiac and is now a part of Clario. We can go to the next slide. The results of the study when they were conducted were reviewed and endorsed by the FDA at the end of 2015. They were then presented to the ICH E14 steering committee, which reviewed the results and accepted them, to then revise the revision 3 for the E14. So now it's accepted to have concentration QT modeling as another methodology to the by time point analysis for assessing QT prolongation risk. We can go to the next slide. This is an example of one of the plots from the iQCRS study. It shows moxifloxacin, which was one of the positive control drugs. And what's shown here is on the x-axis is the concentration in the plasma moxifloxacin in nanograms per ml. The y-axis shows the placebo-adjusted change from baseline in the corrected QTc interval. And as you can see, this is a steeply increasing slope with concentration in the QT interval. At the day 1 concentration of about 1900 nanograms per ml, there is over a 14 millisecond increase in the QTc interval. This is a significant effect, and this is a positive effect drug, which would have a higher risk of inducing torsades de pointes. We can go to the next slide. So I just want to summarize now what was entailed in the revision 3 of the ICH E14 guidance. And this came out with a couple important points. The first one was that you no longer needed a positive control in a QT study now. If you could reach sufficiently high multiples of the exposure, you could negate having a positive control. The number of exposures that you need to achieve was not clarified at this point, but it is now clear that you need to achieve 2 times the high clinical exposure. And we'll talk about that a little bit later on. The second thing is it helped guide decision making. You now have the option to use either concentration QT modeling earlier in clinical development, as well as the by time point or union intersection test. So you have these 2 options, and it provides statistical guidance where the upper bound of the two-way 90% confidence interval could not exceed 10 milliseconds at sufficiently high clinical exposures for a drug to be deemed safe and not have an excessive risk of QT torsades de pointes. You can go to the next slide. Now, at this point, I want to talk a little bit about early precision QT methodology, or EPQT, and why is this so important and valuable? As we discussed, this methodology was used to support the iQCRS study, which then led to the revision of the E14 guidance in 2015. But this is a methodology that is also available to use in the studies that we support, that you're conducting. And this methodology is very important and valuable because it increases the number of data points that you collect. So it allows you to do the assessments in smaller cohorts and early development, and it uses an automated methodology that increases the precision of the measurements. So with higher precision and more data points, you can then use smaller cohorts and early development. You can get a decision on if the drug has safe or not to progress earlier in the decision-making process. As Dr. Ruda said, it's important to have high quality data here. You can also reduce the number of patients you're exposing to a new asset where you don't know the risks of the drug. And it can help reduce costs, since you can do this earlier in development, can determine if you need to progress to later phase trials, and you don't have to do it dedicated through a QT study, potentially get a QT waiver. So there are multiple advantages with the early precision QT methodology for these reasons. We can go to the next slide. What I want to show here on this slide is a comparison of the early precision QT methodology with the traditional methodology that's used for assessing QT intervals. Both methodologies are done using continuous ECGs, where you're collecting the continuous ECG during the 5-minute extraction periods prior to each PTA time point. The data is then transferred to the ECG core lab via the internet to the backend server or on an SD card. But then the methodologies differentiate. With the standard methodology, where you can do this in both patients and healthy volunteers, an analyst will look at the 5-minute windows, and will extract 3 10-second ECGs that have the most stable heart rhythms there. And from those 3 10-second ECGs, they will measure 3 beats and get a total of 9 RRQT pairs. With the early precision methodology, the algorithm will look at all stable heart rates within the 5-second minute extraction window, and it'll extract 10 ECGs that are 14 seconds long. And it will use all the high quality beats within those ECGs. It will then automatically measure the QT intervals and extract all the QT-RR pairs that are available to measure, which typically will come out to be 100 to 120 QT-RR pairs. There will be analyst oversight to make sure that the measurements are done correctly and the quality of the beats is sufficient. But when you have over 10 times as many pairs and data, you now have the higher precision, and you have can use a smaller number of patients. And go to the next slide. This slide really compares 7 studies that were used doing early precision QT methodology, and then we went back and did them using the standard methodology and compared the precision on these 7 studies. And as you can see in both of these slides from the 2 studies that are shown from the 7, the standard deviation using the early precision method was about half what it was with the standard method. We can go to the next slide. When you actually look at the numbers, if we had an assumed change of 3 milliseconds in the QT interval in both studies, the standard deviation was actually about 6 milliseconds with the early precision methodology compared to 10 milliseconds with the standard method. So the standard deviation is about half, which can significantly reduce the sample size that you need to get the data that you need to rule out a 10 millisecond effect. We can go to the next slide now. And just to conclude, what I want to do is summarize what you need to get a QT waiver now by the current guidance. And what the current guidance states is that the QT interval cannot exceed 10 milliseconds at sufficiently high multiples of the clinical exposure. Specifically, the upper bound of the 90% confidence interval can exceed 10 milliseconds at these exposures. And the current guidance says that the exposure has to be twice the high clinical exposure for the regulators to be confident that you won't have a significant risk of Torsades de Pointes. What the high clinical exposure is the clinical exposure of the drug times the one factor that increases exposure by the most, either drug-drug interactions, food effect, renal insufficiency, or hepatic impairment. And this is the one factor that actually we see QT waivers are not accepted by the regulators, because the study does not achieve the sufficient exposures, and they can't be confident that you can get a waiver, and then there's a requirement to do it through a QT study. The regulators were understanding of this limitation, and so in the latest guidance in 2022, an integrated approach is now acceptable, where if you have an in vitro hERG assay, in addition to an in vivo QT study in a dog or monkey that has sufficient margins at the high clinical exposures at those doses in the relevant model, you can then go to the clinical exposure, the high clinical exposure in the clinical study, and still get a waiver. So we'll go on to the next slide. And at this point, I'd like to let Dr. Couder take over and talk about ECG automated analysis and the algorithms that he's helped develop. Thank you.
Thank you, Dr. Patel, and hello, everyone. I'm very excited to be part of this event and to present the most recent technologies we have developed at Clario for ECG analysis. Let's go to the first slide. First of all, I want to introduce iCOMPAS, which is our core technology for QT/QTC interval measurements. What is very unique about this software is that it was designed on data from the International Long QT Syndrome Registry, which is hosted in Rochester, New York at the university. And really, when we developed iCOMPAS, we made sure that we were fitting the software and the development process with this unique set of ECGs from patients that have the various type of LQTS syndrome, various type of genetic disease that led to different ECG phenotype. And it led to a software that was actually learning from a very large spectrum of T-wave morphology and abnormal T-wave, which ultimately led to a software that was extremely precise at measuring the QT. We did then a validation of the software in partnership with the FDA, where we got access to data from 7 thorough QT studies that were hosted by the FDA. And we really compared what was the outcome of the automatic analysis by iCOMPAS and the data that was received by the FDA in these 7 thorough QT studies. The study showed that our automatic analysis was the same than the data that was actually received by the FDA. The next step has been to integrate actually this software into Clario processes and workflow. And basically this technology became the core technology for the early precision QT approach that is currently available at Clario. What I'm going to present today is actually the next step, and what we have been working on, leveraging machine learning and artificial intelligence to somehow improve and enhance what is done automatically on ECGs in these processes. Let's go to the next slide. Before talking about the software and the technology itself, I wanted to introduce the team. We have actually created at Clario the so-called advanced computational approaches to cardiac precision science, and a team that is dedicated to really develop the new generation, the next generation of technology for ECG analysis. This team is unique in the sense that it gathers a set of expertise that goes from machine learning, artificial intelligence, but also integration of software into processes, research in general, and obviously cardiology. So it makes this team very unique, which has developed what I'm going to present in the next slide. So first, maybe the question is, why do we use actually artificial intelligence and machine learning? It is important to understand that today, the technology is not based on linear processing blocks as it was for many years. Now, the machine learning give access to us to nonlinear equivalent, and that enable us to really approach a larger set of problems. What we have decided to work first, as I would call the low-hanging fruits of what we can do with this technology at Clario, is really look at an assessment of the quality of the ECGs that are submitted to the company for analysis. Let's go to the next slide. So when we talk about artificial intelligence, that's a pretty broad set of tools that exist in a sense, and what we have used, it's so-called the neural networks and deep learning approaches. The difference between neural networks and other type of machine learning technologies that it does not require what we call feature extraction. You don't need to do measurement of interval and really characterize the signal to reach the goal you want. You really can just feed the deep learning system with the raw signal, with the ECG itself, and really then target what you want to do with this model. Again, what we basically implemented here, it's a model to quantify even the quality of the ECG signal. And I'm going to give you a little bit more insights about what we do. One of the drawbacks of implementing actually neural networks and deep learning is actually to have a very large amount of data. And this is what we have at Clario. We have millions of de-identified ECG from clinical studies that were all manually reviewed, and they were labeled and can be used now to feed and develop this kind of model. Let's go to the next slide that shows you that we use this deep learning. So we use a convolutional neural network, the so-called CNN, that consists of multi-layer neural networks. And we learn from this large data set we have access and develop this model, so-called deep learning model. Let's go to the next slide. What is very unique in the development of this technology is that once it's created, it is dynamic. There's no reason to actually keep it this way for a long time. The more data you acquire, the more learning you can do. And it's really what we have implemented at Clario, where this model, as you're going to see, is extremely performant, but we continue over time to feed it with new data, and we always try to optimize and maximize the results. The way you do that has to be in a certain way and in a certain framework to avoid any bias in learning from the data and to avoid other problems. What we follow at Clario is actually the framework that was developed and published by the FDA, which is so-called the good machine learning practices that really describe and tells you how you have to develop your model and what kind of processes you need to put in place to make it learning on a regular basis. And this is, again, what we have developed. Let's go to the next slide. Without going too much into the details, I want just to describe very quickly what we have designed. So you have the machine learning. We are feeding this machine learning with what we call ECG epochs that are very short interval of ECG that are extracted from continuous ECGs. They are 3.6 seconds long and basically centered on one single beat. What we have learned with this model is to recognize what we label a good beat versus a poor quality beats. And this is very important because, as I said, this is millions of cases that we have in our databases. And we really we teach the model how to recognize those beats that were manually adjudicated in what we call the 3 bits on lead 2 methodologies, and really recognize those bits that are good quality enough to do a good QT interval and a good analysis. In the opposite, in the other hands, we will also detect the ones that are labeled poor, meaning those actually ECG epochs in which the quality is so low that you cannot actually do a good extraction of intervals. What the model provides, as you see in output layer, is just a score that varies between 0 and 1, where 0 tells you that you have a poor quality signal and 1 that you have a high quality signal. Let's go to the next slide. So we developed actually this model on a set of 350,000 EKGs that were collected from 118 studies. We developed that following, I would say, the standard approach, which is first training, where we train the model to detect this level of quality of ECG. The training was based on 80 studies, including a little bit more than 84,000 epochs, ECG epochs, that were gathered from 929 participants. Then you validate the model, you evaluated the model, and we did that on a little bit less than 12,000 epochs ECG from 74 studies. The validation is different from the test. The validation gives you the opportunity to adjust your model to make it a little bit better on another set of data. But finally, the final step is what we call the test, where we use independent data that the model have never seen. And here we are speaking about 257,000 ECG epochs from 38 studies that were recorded from 1,210 subjects. And really that describes to you the amount of data that was put in place to really develop this model. Let's go to the next slide. And the next slide is showing you the results in terms of labeling ECG epochs. Again, just this very short interval of ECG. And what you can see here is that the performance are quite incredible. The true negative, which is really our ability to detect actually that an ECG epochs contain poor signal, was 93%. Again, in reference to data that was manually adjudicated. And the true positive, which is our ability to detect actually the good quality ECG epochs, was 97%. So very high performance. And again, we compare the AI model assessment to those who made using the standard approach, so-called 3 bits on lead 2. So this is the results for the ECG epochs level. Let's go to the next slide, and I'll show you even more interestingly what happens if we use this machine learning system and we apply it for actually identifying whether there is at least 3 ECG strips per time point available in a whole data set of a study. And we did this evaluation in 3 studies, 2 SAT study and a MAT study. And what you can see in this table is that we had 100% specificity for detecting time points that do not generate sufficient usable replicates. This is very important, right? Because it tells you very quickly if the data that you have submitted have some weaknesses, has actually some ECG that is so poor that you actually cannot do the measurement that you were expecting to do in this measurement. And very importantly, the model did not produce false positive results by labeling time point as usable that would be in truth be unusable for your analysis. And as you see, this is in the table, the last column that shows you the true positive, and you see this percentage are around 99%. So really the model that we have developed not only tells you the quality of short ECG interval, but it also provides very unique data information and insights into ECGs, telling you whether the data you collected during your clinical trial is good enough to do analysis of every time point that you have scheduled to do. Let's go to the next slide. So as of today, we are integrated actually the model into our collect, clean, and store service. Very important, because what's happening is that now that data is flowing into the warehouse at Clario, we automatically analyze all ECG that is fed into our system. It generates a report, as I just said, that lets you know whether the quality of your data is good enough to do the analysis that you wanted to do on this data. So you have a very early automatic assessment of the quality of the collected data from your clinical studies and clinical trials that very soon, very almost, I would say, real time, tells you what is the quality of this data. Again, this is a service associated with the collect, clean, and store functionalities at Clario. Let's go to the next slide. So let me summarize, and again, I want to say that we have developed machine learning application to assess the quality of continuous ECG tracing collected in early phase clinical trial. We leverage this new technology to assess data quality in near real time. With this tool, we can quickly confirm if the data is analyzed and complete in lower dose cohorts. If required, correction can be implemented to ensure optimal data quality in subsequent higher dose cohorts. Our model can also perform a review of the data from collect, clean, and store study, providing you with assurance that the store data will be sufficient quality to support any further analysis. And today, our group is really integrating and developing very novel machine learning technology with a focus on enhancing its service in the management review and analysis of the ECG data. What I presented today is just a small part of what we are doing with machine learning at Clario. We have a set of tools that are currently in development, and that will really define what is the next generation of ECG processes in drug and cardiotoxicity assessment. We are very excited about this, and we will present that in the next series of X talks. So I'm going to stop here, and I would like to thank everyone, and I think we are open for questions.
Excellent. Great. Well, thank you, JP, for taking us through everything there. From the iCOMPAS tool for QT measurement, and then, of course, the AI-enabled continuous ECG quality tool as well. I see we have our panelists back on camera now. I'll take us through some questions that have come in. We actually have a good number of questions, but we also have a decent amount of time, so we should be able to get through most of them. In the event we don't get to your question, of course, we will certainly follow up by email directly with you afterwards. So, first question here looks like probably best directed to Dr. Patel. Do I need to submit a QT assessment for an engineered human recombinant protein? And how about an antibody drug conjugate? So I think the question here really, we've been talking a lot about small molecules, and this is, how does all of this apply to large molecules?
Yeah, that's a really good question, because as I mentioned in the Q&A 3, there was a requirement that large biologics did not need a QT assessment, but the FDA has come back and said that, look, if you have a human engineered protein that has the natural amino acid sequence, then yes, that is not a requirement. But if it has a modified amino acid sequence, they have been looking at this on a case-by-case basis and thinking that you may need to do a QT assessment based on that. As far as an antibody drug conjugate, that's a different situation, because that has a component that's both a large biologic molecule, the antibody, but the payload is actually a small molecule that, when it gets disassociated, it is typically released within the cell, but it could be a small amount of plasma concentration that's also released. So that does need a thorough QT assessment, and you need to do assessments at the right time point to see when the payload is released and how the systemic exposure looks there.
Great, thanks. Second question here, maybe I'll take this one actually. I ran a first-in-human study in the past and am now in phase 2 of development. Can I still use the ECG data from that first-in-human study to support a QT waiver, or will I need a dedicated thorough QT study? Great question. Arguably, actually, the single most common question we receive is I wasn't aware of this at the time I was running my first-in-human. What are my options now? So there's a few considerations. I will say all of this is quickly becomes nuanced, and so it probably requires a bit of a detailed conversation to understand the specifics of your study, and we're happy to follow up. But essentially, there's a few elements in that first-in-human study that have to have occurred at the time of study conduct. The most important of which, the ECG collection had to be matched or time matched to the PK collection. The way that this modeling works that Dr. Patel was describing is the modeling of the relationship between drug exposure, so the PK, and then any change in QT interval. And the only way you can analyze that is if the ECG recordings actually physically occurred at the same time or just before actually the PK draw. So that's prerequisite number one. The other element, though, and because the question asked about supporting a QT waiver versus a dedicated third QT study, the other key element is exposure, right? What was the exposure that was achieved in that first-in-human study that was historically run? And how does that exposure compare to what the clinical dose is likely to be, right? So there's a clinical dose, there's the worst case scenario clinical dose where there's a drug-drug interaction or a food effect. All of these, as I said, this quickly becomes nuanced, but all of these are the considerations. Punchline here, though, there certainly are many case studies we could share in terms of successfully retrospectively analyzing first-in-human study data, assuming some of those prerequisites that I just mentioned had been met, at the time of study conduct. Okay, so I'll go to another question here. This one, Dr. Kuder, I'll send your direction, and it's regarding the AI-based quality scoring system that you were describing. It's kind of a two-part question here, actually. Is it based on the analysis of all leads or just an analysis of a single lead? And then the second question was really what types of ECG sources can be used for the AI-based scoring system?
Thank you, Todd, for this question. So it is, indeed, this analysis is lead specific. So it means that it is actually applied to each lead, and obviously we focus on the lead that are in the protocols in general of the study, but it helps really identifying actually which lead would be the best, right? As soon as you have the quality on all leads, you can really make decision if you see that the pre-selected lead does not have the quality that one would expect. The technology for the second question was developed for Holter and standard 12 lead Holter configuration in terms of lead system. So it is actually applied to this kind of signal. It could be obviously technically applied to other kind of ECG. I'm thinking about ECG, single lead ECG, patch ECG. Yet, I mean, because it's different type of signal, because we know that those devices have usually lower quality in terms of signal, it would probably require to do some kind of initial assessment on this kind of different device collection devices.
Great. Okay, thank you. This is an interesting question. I think I can grab this one. Can ECG data collected at home be used to support a QT waiver? So I think, and this is actually a really good question, because we're constantly thinking about ways to make studies more patient-friendly and address some of the recruitment challenges, and data collection at home is a key element of that. But I think it's important here in differentiating the concept of using ECG data to understand sort of the general safety risks or general safety monitoring versus what's needed in an intensive analysis to support a QT waiver, which is what this question was focused on. So that latter situation does require that the PK and ECG timing is matched, and as I mentioned before in the other question. And so home ECG collection with home PK selection really isn't probably feasible, and also introduces a lot of variables that can affect ECG intervals as well. So for this reason, the data for an intensive QT analysis that's going to be part of a QT waiver, in most cases, will be generated in a dedicated research unit in healthy volunteers, just because of the nature of the data and the data quality that's needed to detect that very small change. That said, there absolutely is a role from a compound safety standpoint for home ECG collection. And that can be via any number of different technologies that have recently evolved, including a simple home 6-lead device, or using a more comprehensive home 12-lead device, which can be done with a visiting nurse, for example. Lots of different ways to approach the desire to add some time points at home versus everything being based in a clinic visit. And mostly those home ECGs and how they're used in clinical development is to assess for any sort of emerging safety signals, right? So to be able to monitor for any clinically important changes in the QT interval, which is very different than detecting these minute changes in the QT interval that are part of a QT waiver strategy. So hopefully that answers that question there. Next one here, I'm going to send to Dr. Patel. This is about patient populations, and specifically the question is, what patient populations can the EPQT method be used with?
Thank you, Dr. That again is a good question that I didn't really clarify during the presentation. But EPQT was really developed, as Dr. Kuder said, on normal ECGs largely. And because of the waveform expectations, we were really using in healthy volunteers There are some patient populations where they don't have much underlying disease, where we can still use it. But the precision and the confidence, the standard deviations get wider with patient ECGs, so you have to use more patients if you use it in patient populations. And so we really use the standard methods more in patient populations where the analysts and the cardiologists can look at the ECG waveforms to really place the calibers more. So thanks for that.
Yep, that's great. Thanks. Actually, maybe one more, Dr. Patel, if I can send this one your way as well. And it's about metabolites. So I'm involved in a small molecule development program that has several inactive metabolites that we expect will be slow to metabolize. Can I still use my first in human study for ECG data to support a waiver?
Again, a very good question. And metabolites are something that I think many sites and developers really ignore or don't think about. And you certainly have to consider the effect of metabolites on the cardiac channels in the QT interval. So if the metabolite's inactive at the drug target, that doesn't mean that it's going to be inactive at the cardiac channels necessarily. So any drug that's a major metabolite, that has a major metabolite greater than 10% of the exposure, those metabolites have to be considered in the QTc assessment. And if they're slow, they have a longer half-life, then we need to design the study to capture those time points and match PK towards their maximum exposure and time. So you certainly can do this in a first-in-human study. You just need to make sure that you capture the time points to match the PK ECG assessments to get the exposure and the relationship, the QT interval, specifically if they're major metabolites, or if the inactive metabolite at the drug target has a known effect on cardiac ion channels or the QT.
Great. Yeah, some great points there. I think I'll just echo what you've said. It's incredibly common for us to hear when we ask about a sponsor about metabolites, oh, I'm not worried about it, they're inactive, and the agency certainly does not view it that way, and the consideration is inactive at your desired target, but not necessarily inactive at the hERG channel or a cardiac target. So good points there, thanks. Let's see, this, JP, we'll send you this one. It's a question about iCOMPAS now. So ECG raw data collected with which devices would feed iCOMPAS by default? And they listed a few here to consider. So regular ECGs, 24-hour ECGs, wearable ECG monitoring devices, or is a dedicated specific device needed for iCOMPAS? So it's data inputs. What can iCOMPAS help with?
So iCOMPAS can read any Holter ECGs. There are some obviously specification in terms of amplitude resolution, sampling frequency, but most serious devices today are fitting to these expectation or technical specifications. So COMPAS was really designed for Holter 24-hour to 72 hours type of continuous 12-lead ECGs. And obviously, in studies, we extract strips from those Holter, and COMPAS also obviously is compatible with this extraction of strips from Holter. So I think it's fair to say that it is for Holter ambulatory ECGs devices as well as standard 12-lead ECG devices.
Okay, great, thanks. And I think there's one last question that I will take here, and then we'll wrap up and follow up with anything further by email. And this is a question about AI. So over time, is the AI-enabled QC tool going to reduce the role of humans in the QC of ECG data? Fantastic question, also a common question. There is a really important concept here, which is the responsible application of AI. There's also the compliance or regulatory aspect of how we can apply AI. And I think the key takeaway here to answer the question is we're not using it in the way that JP described it there. We're not using AI in this use case in order to replace the function of a human, and none of the AI application we're talking about today is being applied in the setting of actual data analysis that would be part of the submission. So what the AI is doing that we were just talking about is helping to improve the quality of data collection as it's being analyzed, as it's being collected at the site, and allowing us a very rapid assessment of ECG quality without needing to take the time for a human to view the entire 24-hour Holter and be able to give feedback to the site to say, we have a quality issue, that we're missing leads, or this looks good, continue as you've been doing. There is a QC process certainly that we implement for all ECGs that are going to be going through any form of analysis, so measurement of intervals, interpretation of morphology. That QC process is not being affected or changed by the use of this tool. It really is about just during study conduct being able to give a rapid assessment of ECG quality. So if an intervention or a change is needed, it's recognized then, as opposed to weeks or months later, or even years later if it's being collected and stored, at which point you have no opportunity to fix it. So great question there, thanks. Corey, I think I'm going to turn back over to you now to close out the session. Again, I thank everyone for joining us and for the incredible engagement there with questions. Again, more to get to, which we will do by email follow-up. And thank you, Dr. Patel and Dr. Kuder, for your presentations today as well.
Thank you.
Thank you.
Excellent. Thank you so much, Dr. Rudo, and thank you, Dr. Kuder and Dr. Patel, for your insight and your input across the presentation and the Q&A. As Dr. Rudo mentioned, if we couldn't attend to your questions, or if you have any further questions, the team at Clario will absolutely have a chance to get in touch with you and reach out to you afterward. But you can also reach out just at the email address there, science@clario.com, and they'll have a chance to continue the conversation after today's webinar. Same goes for if you're someone who's watching the recorded version of this webinar, feel free to reach out and get in touch. I do thank everyone once again for participating today. If you are attending via GoToWebinar, I've added a link in the chat box where you'll be able to view the recording of this event, and you can of course share this link with your colleagues so that they may register to view it as well. You'll also be receiving a follow-up email from XTalks with access to this recording, and after we wrap things up here, a survey window will appear on your screen. Your participation in this survey is always appreciated as it helps us to improve our webinars. Just a quick note, for those of you watching on LinkedIn Live, we do encourage you to create an account on xtalks.com and register for this event, which will not only give you access to today's recording, but to our entire library of content, which will include other webinars, some of which have been from Clario as well. And it's a good time to mention that we do have another Clario webinar coming up that'll be in just a month's time on Tuesday, February 25th. And this one is entitled GLP-1 Agonist Development, Endpoint Strategies to Help Differentiate Your Drug. I have added a link in the chat box to that one as well, but you can of course jump to xtalks.com or use the QR code currently up on screen there to register. You can also join Clario at the upcoming Scope Conference and the ISCE Annual Conference. Just scan the QR codes on the right side of your screen there, and you'll have a chance to connect with Clario as part of these conferences. Before we close off and sign off here today, I just wanted to thank our three speakers once more for their excellent presentation. Dr. Rudo, Dr. Patel, Dr. Kuder, really appreciate your time, really appreciate your insights. We hope everyone found this informative, and we wish you all a great rest of your day. Take care, everyone. Thank you all.
Thanks, everyone.
Thank you.